Hello everyone, my name is Brian Metzger, and today I'm going to tell you about some of the work that I completed during my PhD at the University of Michigan. So gene expression is a central and ubiquitous process in biology. It's a key step in the function for most genes, and within a cell, a large number of components interact and regulate any individual gene's expression. These elements can be broken into two large groups: cis-regulatory sequences, such as promoters or enhancers, which are typically located in the DNA near the gene they regulate, and trans-regulatory factors, such as transcription factors and small RNAs, that are often diffusible molecules that are encoded somewhere else in the genome. Together, the combination of cis-regulatory sequences and trans-regulatory factors, interactions between them result in the regulation of gene expression, increasing or decreasing expression due to some cues. So previous work over the last few years has established that there is considerable variation in gene regulation within a species. So here are some estimates from the literature for a variety of animals, plants, and fungi. So in red, I'm showing the number of genes that are affected by cis-regulatory changes. Then in blue, the number of changes by trans-regulatory changes. And in purple, genes that have evidence of both cis and trans-regulatory differences. Now again, this is all within species. And while the estimates vary considerably, what this shows is that it is not uncommon for a quarter or more of all genes in the genome to be affected by regulatory differences even within a species. Furthermore, any given individual gene's change in regulation is often affected by multiple genes. So this is some work from Alberts et al. Looking at Saccharomyces cerevisiae. And what they find is that somewhere in the range of 6 or 7 genes on average, 6 or 7 loci on average, affect the regulation and expression of any given gene within the species. Now, there are some limits to this prior work. First off, they're primarily pairwise comparisons. And what this means is they can establish the existence of regulatory variation and the number and identity of loci responsible, but they can't determine how variable genes regulation is within a species because they've only looked at 2 particular strains or 2 particular individuals within a species. They also cannot identify the extent to which segregating variation has been affecting a gene has been shaped by natural selection. And finally, they cannot determine the consistency in the number or identity of regulatory variants, again, because they've only compared 2 strains. Now, there are a number of studies that have done multi-strain comparisons over the last couple of years, but in all these cases, the focus for technical reasons has been on cis-regulatory variation. What this means is that to date, there have been no studies focused on trans-regulatory changes and the extent or action of selection on that variation within species. All right. So we have a set of unanswered questions. The first is that we set out to try to address. So the first is, what is the extent of trans-regulatory variation within species? Second, how has this regulatory variation been shaped by natural selection? And finally, how consistent are the number and identity of trans-regulatory variants within a species? So the first question, what is the extent of trans-regulatory variation within a species? So to address this question, we use the yeast Saccharomyces cerevisiae and the promoter from the TDH3 gene. TDH3 is a highly expressed gene involved in yeast metabolism, and we use its promoter sequence to drive expression of a yellow fluorescent protein in order to create a readout of expression. We inserted this construct into the yeast genome, leaving the native TDH3 gene alone and fully functional. We actually did this for a wide variety of Saccharomyces cerevisiae strains. So here is a phylogeny based on whole genome sequencing. We had strains from a variety of different environments as well as geographic locations. So in each strain, we inserted the same construct into the exact same genomic position, and then used flow cytometry with 12 replicates for each strain and roughly 10,000 cells for each replicate. We measured YFP fluorescence. So from this data, we estimated what the median expression was, and then compared that amongst strains that differed in their geographic origins as well as the environments in which they were isolated. What we found is that variation in TDH3 transregulation is actually quite common in Saccharomyces cerevisiae. So here along the x-axis are the names of the individual strains, and on the y-axis is the YFP expression on a log scale. What you can see is that we have considerable power to detect small differences in regulation, and that we actually have considerable variation amongst these strains. And it covers about a 2-fold range from the most lowly to the most highly expressed. So that answers our first question. But given this amount of variation, we want to know to what extent has it been shaped by natural selection. So one of the ways that we can look for natural selection is to actually compare variation that we see out in nature, which has been acted on by selection, to the effects of new mutations, which have not been acted on by selection. That is, the new mutations form the raw material on which natural selection acts, and if we see differences between the effects of new mutations and the effects of natural variation, then we can infer that natural selection was responsible for these changes. So to do this, in previous work, we used the mutagen EMS to introduce many mutations to yeast cells carrying the same TDH3 YFP construct, reporter construct. EMS primarily makes G to A and C to T mutations, and the dose that we used resulted in about 32 mutations per cell. We then, from this, isolated individual genotypes and measured their YFP fluorescence. And so here I'm showing you on the x-axis YFP fluorescence. In black is a set of wild-type controls that did not experience EMS. And you can see they cluster around a narrow range right around 100. And in blue is about 1,500 individual and independent transregulatory mutants containing transregulatory changes. You can see that their variation is considerably higher. So now we have a set of strains that vary in their expression due to transregulation, and we want to compare them to what actually happened in nature. So to do this, we take our distribution of transregulatory mutational effects, and we actually simulate over time what we would expect expression to do if we had no constraints on where it could go. So here I'm showing you 10 different trajectories, all starting from 100%, and how they diverge over time as each individual mutation accrues. Now, of course, we can repeat this many, many times and get a probability distribution. So here, the darker color is high probability, the lighter colors are low probability, given the number of steps that have occurred. And then we can lay over this the changes in natural variation that we see. And the thing that pops out right away is that this natural variation is actually constrained within a relatively small range compared to what was possible or what would be expected given the mutational process alone. And we can apply a statistical test and ask at what point does the natural variation become unlikely given this distribution, and find that that's around 5,000 or so differences. So what this says is that while there is considerable variation in TDH3 transregulation among these strains, that variation is still much, much less than we would expect given a neutral process alone. From this, then, we can infer that natural selection is maintaining the level of TDH3 expression in this narrow range, and anything within that is okay, but outside of that, there may start to be some fitness consequences. And if you're interested in this, there's more evidence for tests of selection as well as some comparisons of fitness in the paper. All right, so now established that there's extensive transregulatory variation within a species, and that this variation has been acted on by natural selection. The final question here is how consistent are the number and identity of transregulatory variants within a species? So to do this, we used a genetic mapping approach. We took individual strains and crossed them to a common reference strain, and then forced those hybrids to go through several rounds of mating or of sporulation and mating. What this does is introduces recombination. We end up with a large number of offspring that all vary in which genomic regions they inherited from which particular parent. We then use fluorescence-assisted cell sorting to take this population and select for either the highest or the lowest expressing individuals within it. And we repeated this 3 times for both high expression and low expression, and then used Illumina sequencing to estimate differences in allele frequency between the low and highly selected pools. So here, as a result of this, we applied it to 3 different crosses where we took different strains from different parts of the cerevisiae phylogeny and crossed them to a common reference strain. So what you're seeing on the x-axis is position along the genome, and on the y-axis is the measure of differentiation allele frequencies between the low and highly selected pool. Each of the little colored bars is a identified loci that shows differences in frequency between the high and low pools. And I think what is immediately obvious is that there's a large number of variants. Right, so we can quantify this. If we actually just look at the number of variants, we see that there's about 100 or so for any given cross. And interestingly, the number of increases and number of decreases, or number of loci that increase and number of loci that decrease expression are about equal. We can further break these individual loci up and ask to what extent are they shared among the different crosses. And what we find is that they're actually rarely shared. So for example, for increases in expression, only 7 of the loci were actually shared among all 3 strains, with another 20 to 30 or so shared on 2 of the 3 crosses, but the overwhelming majority actually unique to any given individual cross, and similar for the decreases. So what this suggests is that there's a large number of loci that are capable of affecting expression. But one concern we had was that maybe our mapping approach just had low power. So we actually repeated the mapping approach for the M22 by BY cross, and we actually found that 75% of the loci we identified were replicated in this independent cross. In addition, of the 8 previously known loci that affected expression of TDH3 between strains that were similar to M22 and BY, we were able to identify 7 of these variants. So this gave us confidence that our approach was actually detecting the loci that were previously known, as well as detecting additional loci that were not detected in previous scans. All right. So from this information, we have a little bit of a paradox in that we have frequent unique changes in regulation, yet selection to maintain the net effect of transregulation. And the solution to this paradox is to actually view many of these regulatory changes as compensatory. So for example, if you started at the center here at some ideal spot, and you could gain a mutation that altered your expression, either increasing or decreasing, and both would be fine. Let's say the one that increased expression was the one that occurred. However, now from this point, however, mutations that subsequently increased even more would then fall outside of some range that natural selection has set, and would not be acceptable, whereas the decreases would be. And this directly produces compensatory alleles in regulation, as you had one mutation that increased expression and one mutation that decreased expression. At the same time, the net change in expression has not changed. Now, of course, you can imagine that from this point, further mutations are possible, and that basically a population might wander throughout these limits kind of haphazardly on a random walk. Of course, however, you could also imagine another population where the individual mutational effect was maybe a little bit stronger after that second mutational effect was a little bit stronger. And what this could do is actually preclude certain steps. And so now you actually have differentiation in the steps that are allowed for any given strain. And so if you imagine this taking place along a phylogeny, you could have many changes in regulation, many independent chains doing the random walking within here. And what this would result in is variation within a range among individuals in the population that are actually masking large numbers of underlying regulatory differences. So this model of rare compensatory alleles may be actually common within species. Recent work from Bloom et al. Showed that rare variants actually contribute disproportionately to quantitative trait variation in yeast. Now, this isn't directly on expression, but this was a range of quantitative traits showing that the majority of the quantitative trait variation was actually arising from variants that were unique or found in only a small number of strains. Similarly, in C. elegans, work from Bernstein et al. In Evolution Letters identified large effect loci affecting phenotypic variation in C. elegans, but these large effect loci were actually located near each other in the genome and had antagonistic or compensatory effects, such that the net change in phenotype was relatively small, even though any individual change could have a large effect on the phenotype. So in conclusion, what I've shown you today is that transregulatory variation can actually be quite common within species, and in fact more common than is previously appreciated. Many of these loci can have an effect on transregulation, and these loci can compensate for each other's effects. As a result, you can have situations where there is underlying regulatory change, yet stasis at the level of actual expression. And that this can all take place in the presence of selection for maintenance of that trait, such that the genetic architecture of a trait can change even while the trait itself is maintained. So with that, I want to thank Patricia Wittkopp, who was my PhD advisor, and particularly Fabian DeVoe,